Good morning! It's February 21st, and this is your daily brief in science. Here's everything you need to know. The Atacama Desert in northern Chile, recognized as the driest place on Earth, faces a pressing water crisis. This region receives minimal rainfall due to its unique geographical position in the rain shadow of the Andes Mountains and the influence of the Humboldt Current. Nearby, the city of Alto Hospicio has experienced rapid growth, with its population increasing from fewer than 100,000 in 2012 to over 143,000. This surge has put immense pressure on the city's dwindling underground aquifer, which has not been replenished in nearly 10,000 years. Residents in informal settlements rely on water delivered by trucks from distant aquifers, a method that is both unreliable and costly. The situation is further complicated by significant social challenges, including poverty and inadequate water supply networks. A recent study suggests that fog harvesting could provide a viable solution to alleviate water scarcity in this arid region. This method utilizes the coastal fog, known as camanchaca, which forms when humid air from the Amazon meets the cooler Pacific air. Fog harvesting involves collecting moisture from fog using fine mesh structures, capturing droplets that can be stored for various uses. According to the study published on February 20, 2025, fog harvesting could potentially supply up to 300,000 liters of water weekly to the informal settlements in Alto Hospicio, significantly enhancing water access. To meet this demand, approximately 17,000 square meters of fog collectors would be needed, while just 110 square meters could suffice for the yearly irrigation needs of local green spaces. The harvested fog water could be used for drinking, irrigation, and local food production. However, successful implementation would require large storage systems and distribution infrastructure. Researchers stress the importance of further studies to validate the model estimates, assess water quality, and identify optimal locations for fog collectors. They advocate for integrating fog harvesting into existing water supply strategies and national water policies, with a detailed fog map for northern Chile set to be released later in 2025 to assist in water resource management. Recent research reveals that for billions of years, Earth's oceans may have been green, largely due to cyanobacteria, before they transitioned to blue around 600 million years ago. A study published in Nature Ecology and Evolution by scientists from Nagoya University highlights that between 3 billion and 600 million years ago, high levels of iron hydroxide in the oceans absorbed blue and red light, resulting in a dominant green light spectrum. Cyanobacteria, the first photosynthetic organisms on Earth, thrived in these iron-rich waters by utilizing pigments called phycobilins that absorbed green light, contributing to the green coloration of ancient oceans. Experiments simulating ancient ocean conditions demonstrated that cyanobacteria adapted to green light grew faster, providing further evidence for the theory of green oceans. Taro Matsuo, a researcher at Nagoya University, emphasizes that this era was crucial for the evolution of cyanobacteria, which significantly increased atmospheric oxygen levels. The proliferation of cyanobacteria played a pivotal role in transforming Earth's atmosphere, making it breathable for other life forms. As these organisms thrived, they produced oxygen through photosynthesis, which reacted with dissolved iron, forming iron oxides that sank to the ocean floor, thereby reducing iron levels in the water. The research also suggests that ancient oceans were likely larger, enhancing their impact on the planet's coloration. Understanding the historical color changes of Earth's oceans can aid scientists in the search for extraterrestrial life, as green oceans may indicate early life on exoplanets. In a potential future scenario, rising temperatures and increasing phytoplankton populations could cause the oceans to turn green again, with studies indicating that 56% of the world's oceans have already experienced a shift in color in the past 2 decades. Recent research has unveiled a promising avenue in cancer treatment through the discovery of tumor-wide RNA splicing aberrations, specifically neojunctions or NJs. These alterations can generate actionable public neoantigens, which may pave the way for innovative immunotherapy approaches. A comprehensive RNA sequencing survey conducted by Kwok and colleagues has identified these neoantigens across various cancer types, indicating their potential as therapeutic targets. Utilizing RNA sequencing data from the Cancer Genome Atlas, the study discovered an average of 94 public NJs per cancer type, underscoring their significance in the realm of immunotherapy. The research identified 315 neopeptide-encoding NJs capable of producing cancer-specific peptides, with a subset showing strong HLA binding potential, positioning them as prime candidates for immunotherapy development. Public NJs were confirmed in patient-derived glioma samples, demonstrating their ability to generate neopeptides that can be presented on human leukocyte antigen molecules. Analysis revealed that IDH mutant gliomas exhibit significantly more public NJs compared to their IDH wild-type counterparts, suggesting a correlation between specific tumor subtypes and NJ expression levels. Intratumoral heterogeneity often complicates the efficacy of immunotherapy, especially in cancers with low mutational burdens. However, NJs present a promising target due to their capacity to produce tumor-specific antigens. The spatial conservation of NJs across multiple tumor regions suggests that targeting these neojunctions could help overcome immune evasion caused by antigenic heterogeneity. This research highlights the critical role of understanding RNA splicing alterations in tumors and their implications for developing targeted immunotherapies. A new study from Mount Sinai researchers reveals a critical link between a patient's exercise capacity during cardiac stress testing and their mortality risk. This research, published on February 19, 2025, in the Mayo Clinic Proceedings, emphasizes the life-saving advantages of maintaining physical fitness, regardless of the severity of heart disease. The study analyzed data from over 15,500 patients over nearly 7 years, examining how various risk factors such as hypertension, diabetes, and smoking affect mortality rates. Lead author Dr. Alan Rozansky pointed out that exercise capacity during stress testing is a more reliable indicator of patient risk than traditional clinical factors. Patients were categorized based on their ability to perform treadmill exercises. Those unable to exercise on the treadmill received medication-induced coronary artery dilation for testing. The findings showed that patients who could exercise had a mortality rate of less than 1% per year, while those tested pharmacologically faced a significantly higher mortality rate. The study advocates for cardiac stress test reports to include assessments of a patient's exercise capability, suggesting that further evaluation should occur if limitations are identified. Dr. Rozansky encourages physicians to discuss the importance of physical activity with their patients, highlighting that even small amounts of exercise can yield significant health benefits. New guidelines are now recommending the integration of short bursts of activity throughout the day to improve cardiovascular health. Mount Sinai Hospital is recognized as a leading institution in cardiology and heart surgery, ranking number 4 nationally and number 1 in New York, according to Newsweek's The World's Best Specialized Hospitals. While the observational nature of the study limits definitive causality, the results indicate a strong protective effect of physical fitness against mortality risk. A new frontier in treating Alzheimer's disease is emerging as NanoNeuron develops biologics designed to cross the blood-brain barrier. This initiative is anchored in innovative technology from Dr. Luciano D'Adamio at Rutgers University, who is a respected figure in Alzheimer's research. Dr. D'Adamio has created a novel molecule, NN-841, which incorporates 2 TNF-alpha inhibitor nanobodies that flank the neurobus. This structure is engineered for effective transport across the blood-brain barrier, a critical hurdle in treating neurodegenerative diseases. Currently, Alzheimer's disease affects over 55 million people worldwide, with projections estimating that number could reach 139 million by 2050. This alarming statistic emphasizes the urgent need for effective treatments. Marco Taglietti, CEO of NanoNeuron, highlights the critical demand for therapies that do more than provide temporary symptom relief, as existing options fail to halt cognitive decline. Dr. D'Adamio believes that NN-841's technology could also benefit conditions related to TNF-alpha, such as traumatic brain injury, multiple sclerosis, and various neurological disorders. Designed to target neuroinflammatory pathways, NN-841, is in the preclinical evaluation phase, with promising results from animal studies showing significant TNF-alpha inhibition in the brain and good tolerability. Nanoneuron aims to revolutionize Alzheimer's treatment, similar to how TNF-alpha inhibitors transformed therapies for systemic inflammatory diseases. Deborah Perez-Fernandez from Rutgers emphasizes the transformative potential of these innovations for families impacted by Alzheimer's. Looking forward, Nanoneuron plans to conduct further preclinical studies and discussions with the FDA, while also seeking additional funding through a Phase 2 STTR grant from the NIH. The neurobus technology significantly enhances the delivery of therapeutic agents to the central nervous system, increasing effectiveness in treating diseases like Alzheimer's. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.